Let us continue with the next very important disease mentioned in chapter 8, Human Health and Disease, that is called AIDS. AIDS, Acquired Immunodeficiency Syndrome. It was first reported in the USA in 1981, and in the last 25 years, almost 25 million people are killed. It is caused by a virus named Human Immunodeficiency Virus, HIV. Now this HIV virus, if you see, it belongs to a group of retrovirus. It has RNA genome with an envelope, protein envelope. Transmission of HIV occurs through various ways, either by sexual contact with infected person, by transfusion of the contaminated blood and blood products, by sharing infected needles, from infected mother to her child through placenta. It is not spread by more touch or physical contact. It only spread through body fluids. So hence infected person should not be isolated from family and society. Student, this disease, you will be surprised that there is no medicine yet for the disease. Almost 40 years of its existence. Let us continue and discuss out who are the people who get high risk or who are high risk individuals with this disease. Those who have multiple sexual partners, drug addicts, taking drugs intravenously, especially with injections, sharing the same needle, individuals who require repeated blood transfusion, children born to HIV infected mothers. So these people are having high, you can say, risk of getting the disease. How it is diagnosed? So diagnosis is done by an enzyme linked immunosorbent assay, that is called ELISA, ELISA, all capital letters. ELISA. E means enzyme, L means link, I means immunosorbent, A means assay. What is the incubation period of it? If you say the incubation period, the time gap between the infection and the first symptoms arrive in the body, it can be from few months to 5 to 10 years. So some people may get the first symptom by say 10 years after the virus has been infected. The most important part of this topic is the life cycle of the HIV virus. Mostly it is asked for 5 months. Sometimes the diagram is given and you have to label out few parts. But if there is a short note, it will be definitely for 3 to 5 months. So we have to understand the diagram. We have to understand the life cycle. So let us start with the life cycle of HIV. Now here I am putting HIV life cycle diagram, how the replication of the retrovirus occur. Now you can see the diagram. So after getting into the body of a person, the virus enters the macrophage. Here the RNA uses reverse transcriptase enzyme and converts itself into the DNA, and then this viral DNA gets incorporated into the genome of the host cell and directs the infected cell to produce viruses. See how clever this genome virus or the RNA of the virus it is. So what does the macrophage do then? The macrophage continue to produce virus particles and hence it acts as a HIV factory. The virus particles enter the helper T TH, that is T lymphocyte, helper T lymphocytes in the blood, where they continue to replicate and produce more virus or viral progenes, and thus the number of the T helper cells get reduced in the infected person. Let us more concentrate on the diagram. If you can see the diagram, the retrovirus, which has a RNA as a genetic material and a protein coat, as soon as it comes near to the cell, the protein coat leaves outside and the RNA genome enters the cell in the cytoplasm of the cell. Now here the genome uses reverse transcriptase enzyme and converts this RNA genome into the DNA. As it becomes DNA now, it can easily incorporate into the host genome. It can cross the nucleus, goes to the DNA of the host and get incorporated into that host DNA. And then it takes the command of the whole cell and asks the cell to produce new virus. So the cell has to produce new virus along with the RNA genome. And once the number of the RNA and this protein coats are produced, ultimately the cell gets killed. So the viruses are released in the blood. Again they infect the other normal cells and this cycle continues. You can see the diagram. Again, human cell, the retrovirus with a RNA genome and a protein coat infects. The coat remains outside, the genome enters, the RNA is converted to the DNA by reverse transcriptase. It enters the nucleus, enters the genome, that is the DNA of the host, commands the host cell to produce more virus progenes and ultimately kill the cell. So with the decrease in the number of the T cells, the immunity also decreases, and the person is unable to produce any immune response even against common bacteria like mycobacterium or parasites like toxoplasma or even some viruses and fungi. Infected cell can survive while viruses are being replicated and released, but ultimately this infected cell die. So is there any treatment? There is no permanent cure of HIV, but there are some antiretroviral drugs. They can only prolong the life, but they cannot prevent the death. Now, if you see about the topic, what the government, what the awareness is being going around. So there are some preventive measures, as there is no cure, but as you say, prevention is better than cure. So National AIDS Control Organization NACO, established in 1991, some NGOs, non-governmental organization, they educate the people about AIDS. World Health Organization, they started number of programs to prevent the spreading of HIV infection, and that includes making blood from blood banks safe from HIV, ensuring the use of only disposable needles, free distribution of condoms, controlling drug abuse, and so things. So students, this is the detailed note on AIDS, life cycle of HIV, treatment and preventive measures. Very important topic. Mostly it is asked for 5 marks, sometimes 3 marks, sometimes the diagram is to be labeled. So definitely you should prepare it. If you have any doubt, you can ask me later in the comment section. Now let us come to the next important topic. Again, this is a disease, cancer. Cancer is defined as uncontrolled division or proliferation of cells without any differentiation. Now what is this uncontrolled division? When a cell does not recognize that how much it has to divide, what time it has to divide, it does not get mature. It keeps on dividing. Such cells are called cancerous cells. So what are the characteristics of cancerous cells? The cell divide repeatedly. They repeatedly divide with uncontrolled cell division. They do not require any external growth factors, so the body nutrition is enough for them. The cancer cell has lost the property of contact inhibition. Very important word, contact inhibition. Now what is this contact inhibition? I will give you one example. Suppose we are playing in a ground and we fell down in a ground and some part of our skin has gone, for example. Now we don't do anything on that part. Say after a few days, automatically the skin comes with the same color, same texture and everything, and the new skin has been replaced with the worn out skin. So our new cells of that skin knows that how much it has to be replaced. Once it touches the old cells, the old good cells, it stops dividing. So as soon as they come in contact, they stop dividing, and they know that now we don't have to divide, the replacement has been done. But when these cells, any of the body cells, loses the memory or property of contact inhibition, so they don't know till when they have to divide. So what they do, they keep on dividing. And as they divide, they form a large mass of cells that is called tumor. So we have heard this word tumor. And sometimes the cell passes out from the tumor to new sites in the body. They are called secondary tumors. So the invasion of cancerous cells from one part to other parts of body by body fluids is called metastasis. Tumors are of two types. Now here you will understand the difference between different types of tumors. Basically we will understand the two tumors, benign tumor and malignant tumor. Normally they ask the question in form of differentiate. So we'll read out the different points between them and we'll understand that. Benign tumor is non-cancerous tumor, while malignant is a cancerous tumor. Benign does not occur or does not show any metastasis. It is non-invasive. It is localized, means it stays at one place. While malignant, it shows metastasis. So some of the cells from one place goes to the another place and again they form tumor. In benign tumor, it stops growth after reaching a certain size, while malignant tumor shows indefinite growth as it has a proliferating cells called neoplastic or tumor cells which grows rapidly and damage other cells or tissues. Benign tumor, it has limited occurrence among the cells of the benign, while malignant there is no adherence amongst the cell. They tend to slip past one another. Benign is less fatal, while malignant is more fatal. So these are the 5 points each you should remember and you should understand that. Now, oncogenic viruses have cancer causing viral oncogenes. Oncogenic viruses have cancer causing viral oncogenes. So what are these? Normal cells of body, they have cellular oncogenes. Yes, normal body cells, they have cells of oncogenes, cellular oncogenes or proto-oncogenes, which are present in inactivated form. So every body cell has cancerous cell, but they are in inactivated form. So they are called proto-oncogenes or cellular oncogenes. But under certain conditions, for example mutation, they get transformed into cancer causing oncogenes and thus the tumor forms. So remember the definition of oncogenes and proto-oncogenes. What are they? Fine. So here we have seen a small introduction of the cancer, characteristics of cancer, differentiate of the tumors. Let us come to the causes of cancer. Now if you see the causes of cancer, the cancer causing agents are called carcinogens, like allergy is caused by allergens, so cancer causing is carcinogens. It can be physical, chemical, biological. If you see chemical agents, it can be some dyes. You might have heard about the when there is a painting, on a wall, they always say, you might have seen in the television also, some ads are there regarding the paints. Alanine dyes are there, and nitrosodimethyl amine. Benzopyrene is one of them. Chemicals in the cigarette smoke is there. These are all chemical agents. While physical agents is ionizing radiation like X-rays, gamma rays. Non-ionizing radiation is UV rays, which can damage the DNA, causing neoplastic transformation. And biological agent, if you see, there are some oncogenic viruses which just I have discussed, and some parasites. So if you see, these are the three different ways or agents which cause cancers. What are types of cancers? So carcinoma, it is a cancer of epithelial tissues, that is skin cancer. Melanoma, it is a cancer of melanocytes of skin. Sarcoma, it is a cancer of mesodermal tissues. Leukemia or lymphoma, it is a cancer of hematopoietic cells, that is blood cells. So 4 types, again, carcinoma, melanoma, sarcoma, and leukemia or lymphoma. How cancer is being detected? What are the methods? So we have different methods, 6-7 methods are there. First is the blood and bone marrow test, just to see whether the number of the cells have been increased or the blood cells have been increased in case of leukemia. Then we have histopathological study, we can call as a biopsy, which we have heard of common. What they do, they take a piece of suspected tissue, cut it, and they stain it, and then they see under a microscope, the pathologists do, and they see the structure, whether it is different from the normal cells or whether it is enlarged and deformed or whatever. Then we have radiography. X-rays are used to detect the cancer. Then we have computed tomography, X-rays to generate 3D dimension image of the internal object. We have MRI, magnetic resonance imaging, again non-ionizing radiation, and very strong magnetic field are used in MRI to accurately detect the pathological and physiological changes in the living tissues. Then we have monoclonal antibodies. Antibodies against cancer, specific antigens are used for detection of certain cancer. Then we have techniques in molecular biology, like it is used to detect genes in the individuals with inherited susceptibility to certain cancer. That is, identification of such genes can help in prevention by advising to avoid exposure to particular carcinogen to which they are susceptible, like tobacco smoke for lung cancer. So these are different methods or detection method of cancer. And last we'll see the treatment. The common approaches is either surgery, radiation therapy, chemotherapy, and immunotherapy. 4 methods. Surgery is just the tumor cells have been operated and removed with help of surgery. Radiation, a lethal radiation of tumor cell is done, taking proper care of the normal tissue surrounding, so that otherwise the normal cell will be cancerous cell. So very proper care has been taken during this method. Chemotherapy, quite common, you might have heard. Cancerous cells are killed by several chemotherapeutic drugs, and these drugs exhibit side effects like hair loss and anemia. And last is the immunotherapy. In this method, the biological modifiers like alpha interferon, which are used, which activates the immune system and help in destroying the tumor as tumor cells. Okay, so and majority of the treatment required combination of surgery, radiotherapy, and chemotherapy. So it is not just one method which it can be used, but combination of 2 or 3 methods is always done. So this is the treatment. So in this case, The topic cancer can be asked into write a short note, which can be 5 marks. They can ask for 2 marks types of cancer. They can ask for 2 marks cancer detection methods. They can ask for 2 marks cancer treatment. They can ask for 5 marks in either of the combination. They can ask for 2 marks the difference between benign and the malignant. They can ask for 2 marks the causes of the cancer, the 3 agents, chemical, physical and biological. And they can ask for 5 marks as a whole. So this is about the cancer cells, cancer topic. If you have any doubt, again you can comment and ask me. The last topic, drug and alcohol abuse, we'll see in the next video, and that will be the last topic of the chapter. So if you have any doubt till today, we can definitely discuss out. You can comment me, you can WhatsApp me on the WhatsApp number. Thank you.